So let us look at bonds in proteins. I guess you basically know what bonds are. When you have atoms being connected, what connects these atoms? Bonds. And therefore, if you have a compound like protein, you definitely have bonds in there because these atoms in this compound have to be connected to one another. So they are connected through bonds, and there are different types of bonds in these proteins. Given the fact that proteins are not all the same, we have different types of proteins, and that therefore means that you definitely have different types of bonds that hold these protein molecules together. So we want to see what bonds are these, which ones are stronger than the other, and can we relate the bonds that we have in these proteins to their different functions. So we start by looking at a scenario here. You have a scientist who is planning to design an enzyme. And one of the things that this scientist will have to think about would be the shape of this enzyme. And that therefore means that they need to know the kinds of bonds that would be in this enzyme. Can you, for example, tell us the different types of interactions that can be found in an enzyme? But of course, for you to be able to do that, you need to know that actually enzymes are actually proteins. So what nature of proteins are the enzymes, and what are the interactions or bonds that hold the different parts of this protein, which is basically the enzyme we're talking about, together. Okay, and why are these bonds very important? And for example, we can note from the question that we have what we call disulfide bonds in proteins. What roles do these bonds actually play? And can you relate that role to stability? If, for example, we talk about an enzyme that is actually stable at some temperatures, that stability could be related to the interactions that they have. And for example, if the interactions that they have or the bonds that they have are disulfide bonds, how does that relate to the stability of that enzyme? And then we also look at the protein structure of the the primary structure of the proteins. Now, how can the primary structure actually influence the extent of folding of the protein? And then what happens if, for example, there is a change in the R group of the amino acid? Can that affect its ability to bond and also function? And if yes, how does that happen? So definitely, we know that proteins are made as a result of polymerization, where you have amino acids combining, and the amino acids basically being the monomers and the protein molecule, which is very large, being the polymer. Okay, so the linkage between these amino acids, these linkages are through what we call the peptide bonds. So the peptide bonds connect the amino acids. What basically happens is that the carboxyl group connects to the amino group of the amino acid. That's where the peptide bond is actually formed. Okay, and the formation is through a process that we call the condensation reaction. So the condensation reaction means that water is usually given out in that reaction. So in this case, yes, there is water that is actually given out. But when we want to break these amino acids into individual amino acids, then we need to use water, and that is what we call hydrolysis. Given that the protein can be very long, it can actually fold. The protein molecule can actually fold, and the foldings result into interactions between the different parts of the same protein. So we have different parts interacting with other parts of the same protein molecule. And these interactions or these bonds that are formed can fall under hydrogen bonds, disulfide bridges, ionic bonds, and also hydrophobic interactions. So we want to know these different kinds of interactions and how they influence the properties of the proteins. So we have already noted that when one amino acid combines with another, they will combine through a peptide bond. So a peptide bond is when you have the carboxyl group of the amino acid combining with the amino group of another amino acid. That's when you form the peptide bonds. These peptide bonds are actually covalent in nature. The meaning of this is that they share electrons. So these bonds are actually between the carbon atom and the nitrogen atom. And usually it results into loss of water. And that's why we say it is condensation reaction. The second type of bond is hydrogen bonds. These bonds are between one electronegative atom with hydrogen. And usually we're talking about oxygen and nitrogen being the electronegative atoms that are bonding with hydrogen. And usually this actually forms what we call the polypeptide backbone. Okay, so those are the two interactions that we can talk about here. The other interactions are the disulfide bonds, or sometimes they can be called the disulfide bridges. What you have here is we have an amino acid that contains sulfur, that is cytine. Okay, so that amino acid called cytine interacts with another. So you have two cytine residues. You have two cysteine residues basically combining, okay, because of the sulfur that they actually contain. And you can see this in this structure here. This is the sulfide, and we have looked at the hydrogen bonds there. Okay, and then we have the fourth type of interaction of bonds. These are the ionic bonds. These ones, ionic bonds happen when you have opposite charges, when you have atoms or ions that have negative charges, opposite charges, one positive, one negative. The bonds, that interaction is what gives you the ionic bond, and that is what we basically see there. And then we have the hydrophobic interactions. Hydrophobic molecules are those that actually hate water. So usually they fall away from water. So you can have protein molecules that are actually hydrophobic because they have hydrophobic molecules. For example, hydrocarbon chains, these are hydrophobic. And therefore, if they have the R group of the amino acid being replaced by hydrocarbon chains, which are very long, that means that this hydrocarbon chain is going to be non-polar, and therefore it is going to be hydrophobic. So you have hydrophobic chains basically interacting, so they are folding away from water. Okay, so with these proteins, they usually fold away from water. That is what you basically see here. And you can see that this is actually falling away from aqueous environment. So you can say that that protein is actually having some good extent of folding. Okay, so you can have peptide bonds for linear chain of amino acids, but for proteins that have undergone some extent of folding, that's when you can see these hydrophobic interactions and also disulfide bridges or disulfide bonds. So that means that if you have folding in a protein, you are likely going to have a very stable protein. So stability increases with the extent of folding. Why? Because you have stronger bonds like the disulfide bonds, which are actually covalent in nature. And usually covalent bonds are actually stronger. Okay, so as a result of that extent of bonding that you can have, you will have different types of proteins because the proteins actually play different roles, and you need some proteins that can easily be dissolved. You have some proteins that need to be very tough. You have some proteins that are actually made up of a few amino acids, and therefore you cannot expect a protein that is made up of a few amino acids to have a lot of bonding, all these different types of bonds that we have. And as a result, we have globular proteins, intermediate proteins, we have fibrous proteins. These categorizations are related to the extent of stability, the extent of strength, and the extent to which they can actually be denatured. Okay, so fibrous proteins are long and insoluble. So this is where you need a lot of strength. So this is where you have keratin, very strong, stable protein, and then collagen. Globular, these are spherical and water soluble, and this is what you need if you are talking about enzymes and antibodies. And then we have those that are soluble but not fibrous, like fibrinogen. Okay, because remember, fibrinogen will turn into fibrin, which has different properties. And then we have very simple proteins. These ones as just see amino acids there. Albumin, globulins, these fall under that. And then we have proteins that have another group attached to them. And this other group, if you have an inorganic group that is attached to a protein, then we are talking about a prophetic group. Okay, that group which is non-proteinous being connected to a protein is what we call a prophetic group. Okay, and then so lipoproteins. This is when you have an organic component being added to a protein. Okay, so lipoproteins, you have a lipid that is actually connected to a protein or conjugated to a protein, and then you have glycogen being connected to or conjugated to protein. That's when you can get the glycoprotein. So the point here is that you have protein molecules that can have other components added to them.